Assalamualaikum and good morning to all viewers. Today I'm going to talk about the methods in gene cloning. As you can see, there are 5 steps of gene cloning. Number 1 is isolation of gene. 2, cleave or cut DNA. 3 is insertion. Number 4, transformation and amplification. And number 5 is screening. The first one is isolation of gene. As you can see, there are 4 types. There are 3. First, bacterial cell, plasmid vector, and DNA gene. At this step, we will choose the bacteria and choose the target gene. It is very easy. And number 2 is cleave or cut DNA. In this step, we will cut the DNA donor and plasmid DNA with the same and compatible restriction enzyme. At the end of this step, you will produce DNA fragments with cohesive ends. Next step is insertion. As you can see, there are 2 types and they will become in one. As you can see, there are 2 types. This is plasmid vector and this is DNA fragment, and the product is recombinant plasmid. This is recombinant plasmid. At this step, DNA ligase is added, and the product is recombinant DNA or plasmid and recombinant non-recombinant DNA or plasmid. And after cut with the same enzyme, some of them will join by base pairing. I will talk one more time. This step is about the insertion of DNA fragments into plasmid DNA to form recombinant DNA. And next, we move to number 4, transformation and amplification. Basically, this step is transformation of recombinant DNA into a bacterial host cell and amplification or recombinant DNA. At this step, we will remove the E. coli and add insulin. As you can see, the star is this: the insertion of plasmid into the host bacterial cell for replication. And number 5 is basically screening. A is antibiotic. There are 2 types, with vector and without vector. As you can see, there are different between them. This one have what we call plasmid, and this we don't have plasmid. And the point is, it will grow if they have ampicillin resistant plasmid, and this will not grow. You understand, right? And B is blue white screening. There are 2 types. They will be blue and some will be white. Basically, will be blue is non-recombinant because it contains intact like Z, and they encode beta galactoside. this, and the white is recombinant DNA. The leg Z disrupted. And that's all for me. Thank you.